introduce our next speaker, Professor Jamie Spangler. So Jamie is currently an assistant professor at the University of Johns Hopkins University in the Department of Biomedical Engineering and Chemical Engineering, where she studies, like most of us here, the impact of cytokine biology on cancer immunotherapy and devising novel engineering approaches to tailor cytokine activity to a cell subset of interest. So Jamie earned her bachelor's degree in biomedical engineering, also at Johns Hopkins, and then she went on to do her PhD research in Dane Wittrop's lab at MIT, where she studied the role of antibody-mediated downregulation of cancer drivers, in this case, EGFR, epidermal growth factor receptors, as a mechanism for cancer therapy. She then moved on to do her postdoc in Chris Garcia's lab at Stanford, where she studied IL-2, and as I mentioned earlier, mechanisms by which you could engineer or tailor IL-2 activity towards cell subsets of interest. So Jamie, thanks for joining us today, and we're looking forward to your talk.
Thanks so much, Jonathan, for this invitation. And this has been a wonderful workshop so far, and really excited to be a part of sort of like-minded cytokine engineering folks here. And yeah, really excited to share some of the work that my lab has been doing on engineering interleukin-2, which I know has already been a hot topic today. And so fortunately, since such a wonderful background was already given, I won't have to go too much into detail. But we're going to talk about some different strategies that our lab has used to make that into a targeted cancer immunotherapy. So as you guys are familiar with from the talks earlier, but just to kind of introduce the molecular scale here, we have interleukin-2 was actually the first cytokine to be both genetically and structurally characterized. Structure was characterized by Chris Garcia's lab back in the early 2000s. And since the structure was characterized, what was recognized is that, or what was known about IL-2 is that, of course, it has the intermediate affinity receptor, which consists of just the beta and gamma common gamma chains. But it also has the high affinity receptor, which also includes the alpha chain that serves as sort of a sponge, increasing the affinity of IL-2 for the overall complex. And essentially, as a result, cells that are low in IL-2 or alpha have a 1 nanomolar affinity, whereas there's a 100-fold higher affinity of 10 picomolar for cells that express high levels of the IL-2 or alpha receptor. And as you know, effector cells in their naive state, things like CD8s, CD4 effector T cells, as well as NK cells, do not express high levels of the IL-2 or alpha receptor, whereas regulatory T cells express constitutively high levels of IL-2 or alpha, although that balance shifts when effector cells become activated. But importantly, right, IL-2, right, has played a role in both the cancer side and for infectious diseases in terms of its immunostimulatory potential as a growth factor for effector T cells. But its role in regulatory T cells has actually become increasingly important. And my lab also does a lot of work in harnessing IL-2 as a Treg activator. And IL-2, actually natural IL-2, and especially at low doses, can actually serve a lot of therapeutic purposes in autoimmune disorders and for transplantation medicine by just having its natural proclivity towards cells that express the alpha receptor, so Tregs. But we want to actually flip that, right, in the case of cancer, to turn IL-2 into a powerful anti-cancer therapeutic. So I'm going to talk about two different strategies that my lab has been involved with. The first is engineering a cytokine antibody fusion protein. We've actually done this on both the effector and regulatory T cell sides. And today I'm obviously going to focus on the effector side for cancer. And then I'll also talk about the development of cytokine mimetics, essentially to using computational approaches as potential anti-cancer therapeutics. So starting on the anti-IL-2 antibody approach, as we know, right, IL-2 sort of hangs in the balance between activating effector cells and regulatory T cells, and it would be ideal, right, to have a molecule that could bias it towards one or the other. And it turned out, as was discovered by Onur Boyman when he was in Jonathan Sprent's lab back in 2006, was that there's certain antibodies that actually just were used as Western blotting antibodies, one known as S4B6, which bias toward effector cells, and a complementary one known as JES6-1, which biases towards regulatory T cells, that were known to basically influence the effects of IL-2 in different directions, overall manipulating the balance of immune cells to create either an immunostimulatory or immunosuppressive environment for various therapeutic purposes. But the question, of course, was what is the molecular basis, right, for this biased immune cell subset activation? So to get to the bottom of that, in my postdoc in Chris Garcia's lab, as Jonathan mentioned in the introduction, I actually solved the crystal structure of the S4B6 and JES6-1 antibodies bound to IL-2. And as you can see, JES6-1 and S4B6 actually binds to opposite sides of the IL-2 molecule, which is very interesting and appropriate because they're actually having opposite effects on the overall mechanism of IL-2 signaling. And when you notice specifically about the S4B6 antibody is it's completely overlapping with the IL-2 or alpha subunit, right? It's sort of taking the place of IL-2 or alpha and blocking it. And it turns out it's also actually kind of potentiating the activity of the IL-2 cytokine by making it higher affinity for its beta and gamma receptors. And JES6-1 is on the opposite side, actually blocking beta gamma, and has more of an effect, but not seeming to affect alpha and having more of an effect on regulatory T cells. But focusing in again on the S4B6 antibody, which we see is sort of overlapping in a large part with the IL-2 or alpha, it turns out that S4B6 IL-2 complex can actually stimulate, of course, both effector and regulatory T cells, but its effects are differential. So if the S4B6 IL-2 complex encounters an IL-2 or alpha low effector cell, as I mentioned, the S4B6 actually poses IL-2 in a conformation such that it's able to readily engage the beta gamma receptors and has a higher affinity for engagement. And that allows for strong signaling on IL-2 or alpha low effector cells. However, if S4B6 IL-2 encounters an IL-2 or alpha high regulatory T cell, it turns out the IL-2 or alpha, right, is blocking the epitope where IL-2 would normally bind to that receptor. And this allows for the S4B6 IL-2 to still form the beta gamma complex, right, the heterodimeric complex on the surface of regulatory T cells. But now, right, the game has changed, right, because the Tregs no longer have that 100-fold advantage that they originally did. And because the IL-2 or alpha low effector cells are overall more abundant in the immune system, and because they express higher levels of beta, there's going to be an overall proclivity towards activation of effector cells over regulatory T cells. Again, regulatory T cells can still be activated through this receptor, but they've sort of lost their edge in that system. And so that was sort of the mechanism behind why the S4B6 antibody was biasing the IL-2 effects towards effector cells. So this was all well and good, and it was actually shown in a number of different cancer models that this could be an effective approach for cancer immunotherapy, but unfortunately things fall apart. And the same is true for the S4B6 antibody IL-2 cytokine complex. And the question would be, right, if we were going to develop this therapeutically, complex dissociation would cause two different problems, right? One would be the pleiotropic effects, right, because IL-2 would no longer be biased in the way that it originally was towards effector cells, minimizing activity on Tregs, but also rapid clearance, right? Because the antibody through FcRn-mediated recycling was giving all the advantages, right, than antibody would in terms of the serum half-life, which would all be obviated and eliminated by the dissociation of IL-2 from the antibody. There's also issues with dual construct dosing, which can complicate the optimization and downstream development pipeline of the of IL-2 antibody complex as a drug, and increased regulatory hurdles like having to show sort of stoichiometrically, right? What if we add excess of IL-2, excess of the antibody, but then you have free IL-2 or you have free antibody, or you have unsaturated antibody, there would be a number of different issues related to that. So as I foreshadowed before, we said, why don't we put 2 + 1 together and make 3? And make something better. And that's exactly what we did. And we made an immunocytokine. So when people probably think immunocytokine, right, normally you're thinking about the antibody, right, targeting a particular antigen, potentially in the tumor microenvironment, and then dragging the cytokine sort of along for the ride, piggybacking. But this is actually a little bit different, where we put it at the N-terminus and actually made it into an intramolecular immunocytokine, where the antibody is actually binding to the cytokine itself and directing that towards the cells of interest. So the first thing that we wanted to do, again, towards with an eye towards translationally, right, turning this sort of stable, stoichiometrically balanced molecule into a potential drug, was to actually turn it into an anti-human IL-2 antibody. So the S4V6 antibody is against mouse IL-2, but does not cross-react strongly with human IL-2. And so instead, we use the 602 immunocytokine, which was also discovered actually by Onur Boyman, as a sort of analog of the mouse S4V6 or anti-mouse IL-2 S4V6 antibody, and acts through a similar mechanism. And we wanted to first show that the molecule was actually behaving in the way that we thought that it should behave. And to demonstrate that, we actually performed a series of different bilayer interferometry binding studies where we looked at binding of IL-2 to either the free 602 antibody shown in gray, or to the immunocytokine, which is intramolecularly assembled. And you can see that if you get to really high concentrations of the IL-2, you can start to displace the IL-2, the 602 immunocytokine bound, intramolecularly bound IL-2, but overall the significant reduction in the EC50 of binding, or sorry, in the KD of binding was indicative of an intramolecular interaction. Further to show that, we wanted to show that we were blocking the interaction of the IL-2 or alpha subunit with the IL-2 by binding intramolecularly to the immunocytokine. And indeed, we see a complete ablation of binding to alpha. We also thought that we might be potentiating, right, binding to beta, as happened in the case of S4V6. And indeed, that's what we saw compared to just free IL-2. We have significantly improved binding, both due to the bivalency of the molecule, but also due to enhanced interaction with the beta receptor. So that was exciting. But the important thing that we wanted to see was that we could actually bias towards cells that express IL-2, that do not express IL-2 or alpha, versus those that do, as is true with the S4V6 antibody. And the way that we see that, right, is that when the S4V6 complex, so consisting of the antibody and the cytokine, are compared to IL-2, they're pretty much the same on effector-like cells, right? Because there's no IL-2 or alpha, but there's a significant impairment of that signaling, right? A significant right shift in the EC50 when we have S4V6 complex interacting with a cell that expresses IL-2 or alpha at high levels. Unfortunately, this allows for, again, selectivity towards effector-like cells. But unfortunately, the same was not observed actually with the 602, with the anti-human IL-2 antibody. So we had to turn to some engineering as a mechanism to actually restore that bias. So the way that we engineered that was we put the 602 scFv, variable heavy, variable light, in that order on the surface of yeast. We then made a mutagenic library in which we mutagenized, randomly mutated CDR1 and CDR3 of both the heavy and light chain, transformed that into competent yeast. And when we did the selections, we were actually looking not only for things that bound, but actually for things that would basically right shift this curve, essentially, so that we could get things that would be more competitive and more effective at binding in the presence of the IL-2 or alpha subunit. So we did selections for IL-2 in the presence of IL-2 or alpha to see things that would resist competing with IL-2 or alpha and thus be more effectively competing with IL-2. With the receptor in terms of the antibody cytokine interaction. And so long story short, we were able to evolve clones that had not only higher binding, but also had right shifted, that curve. And you can see that right shift if you actually normalize of about 10 to 50 fold. And ultimately, what we wanted to see was, could we actually use that to manipulate the Treg to effector cell bias? Now, remember, IL-2 by itself is biased toward Tregs actually by a 40-to-1 ratio. The best we could ever do would be 1-to-1 ratio, right? And that could be achieved if we got exactly the same signaling on effector cells as we got on cells in the presence of CD25. And what we found was we had a range of different variants, including our lead variant, which we call F10, immunocytokine, which had about a 1.2 to 1 bias in favor of Treg. So very, very close to an even playing field. And ultimately, whereas again, the 602 complex had very little bias towards the IL-2 or alpha, towards the effector-like cells, we were able to reintroduce that bias where we lost we lost significant activity on the Treg-like cells to ultimately favor the effector-like cells for the F10 immunocytokine. And that actually translated when we took these into human PBMCs and looked at CD8 positive T cells. Same was true as per CD4 positive T cells. Against regulatory T cells, you can see that there was a significant advantage for the F10 immunocytokine, whereas it lost a little bit of activity on the CD8 positive T cells for this F10 immunocytokine formulation. We saw a much more significant 5-log drop in the F10 immunocytokines activity on regulatory T cells, which led to an overall advantage for CD8s as well as CD4s effector cells compared to Tregs in terms of their activity ratio and in vitro. So ultimately, we wanted to see would that actually translate into an activation or an overall expansion in vivo. And indeed, in mice that were dosed with either IL-2, the 602, IL-2, 602 cytokine antibody complex, or the F10 immunocytokine altogether, we were able to see a significant increase and expansion of the CD8s at the expense of Tregs, as well as NK cells at the expense of Tregs, whereas we saw pretty much no change when we added the complex. So there was actually an advantage of doing this as an immunocytokine. And indeed, that translated into a reduction in tumor growth in 2 different models. We did a B16F10 syngeneic model of melanoma, and in mice that were dosed, we saw a significant reduction in tumor growth over the course of treatment. We also performed a colorectal cancer model, specifically the CT26 model. And again, we saw a slowing of the tumor growth compared to, in the first case, we compared it to just free IL-2, but that's not super fair comparison since IL-2, clears very quickly. So we created this control immunocytokine that was essentially IL-2 fused bivalently to an irrelevant antibody that it does not bind. So when you have just IL-2, it has seemingly no significant effect in the particular model that we had treated for this dosing strategy, whereas we were able to see significant tumor growth reduction in the case of the F10 immunocytokine. So we're building on this initial promise to other tumor models and to combination strategies as well. And look forward to seeing this, sort of develop as a story. The second story that I mentioned is a computational design story, which was performed in collaboration with David Baker's group and has been ongoing also since my postdoctoral studies, and really is, using this concept of how can we flip the bias of IL-2. We talked before about, the way that we can do that using an antibody that binds to and manipulates the interactions of IL-2 with its receptors. But how about just manipulating the IL-2 itself in terms of its interactions with receptors? And that's been done before using muteins and other approaches that are similar. But what if we just wipe the slate clean, right, and start from start from scratch and see how we can build sort of an ideal IL-2? And what an ideal IL-2 would look like, right, in theory, one would imagine would have essentially no interface with the IL-2 or alpha, right? Scrub that interface away and have an enhanced interface, an enhanced binding to the beta gamma subunits. So what we did in collaboration with David Baker's group was we focused on these bottom 3 helices shown in yellow, blue, and red here, and sort of rebuilt this green helix. So we isolated the interactions that were relevant to forming the shape and complementarity of the cytokine interaction with these beta and gamma chains. And then we sort of isolated those in 3-dimensional space and then built piece by piece, right, essentially a 4-helix bundle that not only, right, would recapitulate those interactions without the interactions originally shown with the IL-2 or alpha subunit, but also for enhanced stability. To really pack the core of that 4-helix bundle so that we could have a much more stable version of the IL-2 cytokine, which would be beneficial for both downstream development, for therapeutic sort of translation, and also potentially for other applications in terms of overall stability of the molecule. And in addition to impacting on that hydrophobic core and enhancing the overall structural properties of IL-2, we also said, well, we can also just make up a new topology, right, for IL-2 in this Neo2 format, which is what we're calling our IL-2 mimetic format. So you see we've rearranged the helices in some way, but what we've also done is, whereas the IL-2 cytokine, right, in nature, just like all other cytokines in nature, has the conformation of what's called up, up, down, down, meaning that from N to C terminus, the first two helices are facing up, the second two are facing down. We said, well, that actually leads to these really long intramolecular loops, right, from the top of the first helix to the bottom of the second, or the bottom of the third to the top of the fourth. And why not just make those actually really short, compact connectors by going up, down, right? Sort of alternating between the different directionalities of our secondary structure and our helices, so that we can actually just get those nice short connectors and ultimately get rid of a lot of that extraneous spaghetti that could be destabilizing or manipulating the protein in other ways. So what we did in collaboration with the Baker Lab is they popped out a bunch of designs computationally, and then we put them into, they were put into yeast display system, where we essentially selected against the heterodimerized through leucine zipper, actually beta gamma version of the beta gamma subunits together. We selected for things that bound with high affinity. After multiple rounds of selection, we popped out this Neo2 molecule that essentially has no interaction whatsoever with the IL-2 or alpha subunit, which is exactly what we want, and has a left shifted or enhanced interaction with the beta gamma subunits compared to IL-2. We also showed that this resulted in actually left shifting the curve for IL-2 or alpha negative effector-like cells, and having no effect, of course, for regulatory T cells compared to IL-2, since it's essentially the clean version, right, of what we had seen before in terms of IL-2 or alpha interaction, since there's absolutely no engagement of IL-2 or alpha on the part of Neo2. This ultimately results in an enhanced binding or an advantage for effector-like cells. And in addition, as I said, there were multiple efforts made to structurally stabilize the Neo2 mimetic. And as a result of that structural stability, we were able to demonstrate that the protein became significantly more heat stable. This is looking at two different lines of evidence. First, just looking at the binding of Neo2 after being heated for various periods of time at 80 degrees Celsius, and then almost boiling the cytokine to 95 degrees Celsius for a period of 1 hour. We were able to see that there was no functional stability loss for Neo2, whereas the IL-2 parent cytokine was dead in the water. So ultimately, we wanted to see if that would lead to an enhancement in terms of tumor control and potential cancer therapy. So we looked in a CT26 model, once again, of colorectal cancer, also in a B16F10 melanoma model. This is in combination with the TA99 antibody to drive immunotherapeutic effects. And what we saw, again, was a reduction in tumor growth for the Neo2 compared to PBS treatment or just IL-2, the parent molecule, increased overall survival. We saw the same increased survival and also a significant reduction in toxicity resulting from the Neo2 administration compared to IL-2, whereas half of the mice in the IL-2 TA99 group had to be sacrificed due to the toxicity alone, and the other half died due to the tumor. We actually had 50% long-term survival after 8 weeks in the B16F10 melanoma model, work that was done by Mike Dugan at Harvard Medical School. And so the gotcha question that we always get is, of course, will this be immunogenic? And the question is, one could argue, may be immunogenic, may not be immunogenic. But the promising effects that we saw in this initial work that was published in 2019 was that there was no effect or there was no apparent immunogenicity after biweekly dosing for 28 days, which was an encouraging result for this molecule. But building off of that molecule, right, there's different directions, as I said, that we could go. And we had actually stabilized this molecule so much, right, so much structural stability, that we could actually split it into its component helices, which was a really exciting sort of finding, that we could actually break up protein secondary structure, and then we could actually reconstitute that in a conditionally activatable way. So conditional activation, right, is very exciting because most tumor markers suck because they're expressed on cancer cells, but they're also expressed on a lot of other cells. So an approach, that you can use to get more specificity would be to have, instead of targeting a single marker, to actually target 2 different markers, so that when you have an antibody or some sort of binding moiety against marker 1, but not only that, you have also a second antibody or binding moiety against a second marker, then you can reconstitute the cytokine on the surface of that target cell to then learn an immune cell that can ultimately kill that target cell. So this would be a promising approach, right, for dual expressed antigens or co-expressed antigens in cancer. The first thing that we wanted to do for proof of concept that this molecule could actually be created and could be split, and this is actually described in more detail in a Nature Biotechnology paper that came out literally yesterday. So I encourage you all to take a look. And in collaboration with David Baker's group. And we split it up in 3 different ways, looking at the helix 1 plus 3, 2 prime 4, 1 and 3 plus 2 prime 4, and then 1, 3, 2 prime plus 4. And ultimately trying to see how can we actually get the different splits not to activate, but get the molecules get the splits in combination to be active, but of course less active, right, than the intact protein, so that there's still room to grow in terms of the targeting. And so indeed, we saw that we were able to achieve functionality with all 3 split conformations, but it was only this split conformation H1 plus H3 2 prime 4 where we were able to have minimal activation of the individual splits themselves. So we chose the H1 plus 3 2 prime 4 to move forward with, and then we conjugated it to DARPins against HER2 and EGFR, 2 representative potential cancer markers. And we took target cells that co-express HER2 and EGFR compared to cells that express neither target. And we looked to see whether we could actually achieve trans signaling in this sort of mock system. And long story short, we were able to achieve that pretty exclusive signaling on target cells with minimal activity on off-target cells. We also saw that if we used just one split or the other, we were not able to achieve activation, whereas in combination, the 2 splits together were able to achieve robust trans signaling. But downstream, we wanted to see if that trans signaling could then translate into efficacy in a tumor model. And so to that end, we looked at 2 different models. We first looked at a PD-L1 high model of B16F10. And in this particular model, we used an anti-PD-L1 nanobody that was conjugated to both versions of our split, H1 and H3 2 prime 4. And what we saw is that when we had control splits fused to an irrelevant antibody, they behaved pretty much the same as either PBS or TA99 in terms of tumor growth. We saw some efficacy of Neo2, the intact cytokine or cytokine mimetic itself, but then the Neo2 splits actually led to the best reduction in tumor growth and the most extension of tumor survival, including 1/5 of the mice that were cured. And you can actually see, in addition, that the toxicity, there was a huge differential, whereas the Neo2 splits plus TA99 did not show any toxicity due to their conditional activation and their superior tumor targeting. We did see some toxicity, particularly towards the end of the experiment for the Neo2 group. The next thing that we wanted to show was that not only could we target a single cancer antigen, but of course the advantage here was that we could target 2 different antigens at the same time. So in this experiment, we looked at HER2 and PD-L1. And indeed, when we have the anti-HER2 plus anti-PD-L1 Neo2 splits, we get the best tumor control and reduction in overall tumor growth. We still get an effect again from the anti-PD-L1 Neo2 splits happening on the target cells recruiting in an immune cell. But the overall performance is superior for the anti-HER2. And the important thing also was in the Neo2 group, whereas we did see a significant response, we also saw significant toxicity, again, because we lacked that sort of conditional targeting situation. And 3 of the mice in that group had to be sacrificed due to weight loss, whereas we had long-term survivors in the case of the splits being used, either complementary splits or identical splits. And then finally, this same method and same approach can also be used for cis targeting in the tumor context, in addition to trans targeting. So we can imagine, right, using different splits against a target on the immune cells or on T cells themselves. In this case, we had anti-CD8 to drive activation of CD8 positive T cells to ultimately kill target cells in the tumor. And you can see that there was an increase if you compare control Neo2 or control splits to the anti-CD8 targeted Neo2 or anti-CD8 targeted splits. You see increase in the CD8 positive T cell to Treg ratios in both the lymph nodes and spleen. And that ultimately translates into superior control of tumor growth for the Neo2 splits in combination with the TA99 antibody, both in terms of tumor growth and survival. As you know, and as we'll discuss further, and I'm looking forward to in the panel session and beyond, there are many, many different molecules sort of in development. So this is only 2 small examples of molecules that are in a much larger landscape, but we're very excited about the potential for these different approaches in cancer immunotherapy. So to just summarize things, sorry, I'm probably running over here, but just quick wrap up. These cytokine antibody fusion proteins have a lot of value as delivery agents for IL-2 therapies and can realize its cancer potential. Their computationally designed de novo cytokines also have a lot of potential to increase the stability, selectivity, and ultimately the therapeutic efficacy of molecules. And these split de novo mimetics allow for conditional activation to specifically target the tumor microenvironment. I'd like to end by thanking the amazing people who are behind this: Jacob Tamala and Alyssa Leonard, 2 phenomenal postdocs, as well as Lin Yang, a phenomenal grad student, master's students and undergrads who also helped out. Amazing collaborators, David Baker, who is the force behind the IL-2 mimetic story, as well as Michael Dugan, who was involved with that story, and collaborators Mark Kovar and Warren Leonard on the antibody cytokine fusion story. Thanks to funding sources. Thanks to all of you for your attention. Wow, I'm wearing the same shirt. I really should be more original. Have a great day, and looking forward to further discussion.
Thank you, Jamie, for that great talk. Really cool to see the split Neo2 approach. A nice additional application of the original publication. Really nice. I think in the interest of time, let's just take one question. We'll take a short break and then come back for the panel, where I think we can cover probably a lot of the other questions that will come up, particularly that last slide you showed, which I'm sure will generate a lot of discussion. But we have one question from Amin that's basically a little bit in line with that last slide you showed, around what your thoughts are on the activity of IL-2, the anti-cancer activity of IL-2 muteins that are alpha beta biased, as is starting to emerge from both Chris's lab and Cytokine, and the beta gamma biased approaches.
Yeah, no, it's a great question. And yeah, like there's such a huge landscape, that's sort of being developed exactly around the sort of alpha beta versus beta gamma, and on both sides of the coin, right? Using these, both in the cancer landscape and the autoimmunity. I think that for me, what I really love about the antibody-based strategy, going back to the first part of the talk, is you have that extra layer of control. So essentially, when you make mutations to the IL-2 itself, then your IL-2 is already determined in terms of what its binding and activation properties are going to be and how those are going to be biased. When you have like another molecule that's essentially like a mask in terms of its binding and its epitopes, you can actually reveal, right, selective parts of the IL-2, but you don't lose functionalities, right? You don't lose certain properties of that molecule. So I think there's certain aspects, right, that are beneficial and certain aspects that are not beneficial. But what I think is useful about having an antibody sort of selective or driven approach is that you allow for sort of selective exposure of different parts of the molecule at particular times, which I think is an interesting approach. And yeah, I think the other ones are great because they're sort of like a built-in sort of functionality, but it does make the molecule into a little bit of a determined kind of one-trick pony, because it won't be able to exhibit those other functionalities, which may be useful at a later point.
Great. Thanks, Jamie.